Now that we've looked at the heart and the movement of blood through the heart, let's look at the vessels that the heart is connected to. We're going to talk about the circulatory system. We're going to look at the histology of the vessels, and we'll talk about how the system is regulated and how blood pressure is regulated. We're not going to get into the names of all the different branches of the circulatory system. That's something you'll hopefully enjoy doing in the lab. We'll start with the general anatomy, the general layout of the system, and we'll also look at the histology of the tissues. We'll do that in part A, and then in part B, we're going to look at the physiology and regulation of blood pressure and of the cardiovascular system itself. I love these couple of images here. These are from the exhibition known as Body Worlds, and what they've done is they've taken real human cadavers and they've injected latex, so this rubbery substance, into the circulatory system. After that, they've used enzymes and chemicals to remove all the flesh. So we're left with this cast of the circulatory system. You can see how fine the vessels are and how many vessels there are in the face and also in the lungs and in the liver. In this case here, they've used a very liquidy, very fluid latex, and this runny latex has gotten into all of the capillary beds. Looks like you're seeing a solid dog, but if you look closely, you can see he's kind of spongy looking. You are seeing the blood vessels in the dermis of the skin. Let's start with some terminology. Large blood vessels that carry blood away from the heart are known as arteries. Usually they contain oxygenated blood, but as you remember, that's not always the case. So the pulmonary arteries that lead from the heart to the lungs contain deoxygenated blood. Arteries are going to branch into smaller vessels called arterioles, and the arterioles will lead into a capillary bed made up of capillaries. Capillaries are microscopic vessels, and this is where exchanges of gases and waste and nutrients occur. The blood is going to be collected into venules, and then the venules are going to lead into veins, and veins carry blood back to the heart. The blood that's found in a vein is usually deoxygenated, but not always. So the veins that come back from the lungs to the heart, the pulmonary veins, they contain oxygenated blood. The other thing to note is this is a typical capillary bed that we're seeing here. This is different from a portal system. Remember the hepatic portal system? Well, in a portal system, we have two capillary beds that are connected to each other. Blood passes from one bed to another without going through the heart. The capillary beds that are connected as part of the hepatic portal system, they're connected by a blood vessel called a vein. So in that peculiar case, the vein is not leading to the heart. Arteries and veins differ in their histology. Arteries contain blood that's under a lot of pressure. Blood is being forcefully pushed away from the heart. After the blood travels through a capillary bed, it loses most of that force. It loses that pressure. So veins are much thinner walled. They don't have to withstand high pressures. Arteries, on the other hand, have to be more structurally sound. They have to remain open at all times, and they have to be able to put up with that pressure. Capillaries are very thin walled. They're made up of a single layer of squamous cells. This is a layer known as the endothelium, and then they simply have basal lamina, so a thick extracellular matrix and layer of collagen around the outside. Note in the capillary bed there's something called a through-fare channel. We will come back to the function of that in a little while. In both arteries and veins, we have three tunics, or layers. We have the tunica interna, we have the tunica media, and we have the tunica externa. The space within the middle of an artery or vein is known as a lumen. And we have a thin layer of endothelium that's in direct contact with that lumen. The endothelium is only a single cell thick, and it's made up of these flattened squamous cells. Remember that epithelial tissue is always connected to a basement membrane made up of collagen and other fibers. An endothelium is simply an epithelium that's exposed to an inner lumen. Underneath the basement membrane, but only in arteries, we have what's called the internal elastic lamina. And it looks like a piece of Swiss cheese in this diagram. It's made up of elastic fibers and collagen fibers, and the holes allow material to pass through more quickly. Outside of that, we have the tunica media, which is made up of smooth muscle. And note that it's much thicker in the artery than it is in the vein. It provides quite a bit of support for the artery. Outside of that, we have another elastic lamina, the external elastic lamina, and once again, it's filled with holes to allow material to move, but at the same time, it's a structure that offers support while still allowing for some stretch. These elastic layers are going to push back against the pressure that builds up in the lumen. Finally, outside of that, we have the tunica externa, which is connective tissue consisting mainly of collagen. And one last thing to note, look at the vein and you'll notice that there are little cusps of endothelial tissue that form valves. And that prevents the blood in the vein from flowing in the wrong direction. In the bottom photograph, you're seeing an artery and a vein as they appear under the microscope. They're passing through adipose tissue, fat tissue. Notice that the artery has a circular cross-section. That thick layer of muscle and also those elastic layers help it maintain its shape. Veins, on the other hand, tend to collapse, as you can see. They tend to be far more floppy. At the top, you're seeing a diagram that shows a blood vessel known as the vasa vasorum, the vessel of the vessel. So you can see that we have these little tiny blood vessels here and here in the tunica externa of a large artery, and we also have them in the tunica externa of a very large vein. These blood vessels distribute oxygen and nutrients and pick up waste and carbon dioxide from the muscles, the smooth muscle cells, for instance, that make up the blood vessel. So blood vessels, if they're big enough, have their own blood vessels. The arteries that leave the heart, the aorta for instance, are known as elastic arteries. They're very thick-walled, and most of that thickness comes from the tunica media. Mixed in with the smooth muscle cells of the tunica media are sheets or lamellae of elastic fibers. Elastic arteries lead into muscular arteries. These are thinner-walled. The tunica media is thinner. The tunica media also doesn't contain as much elastic fiber. Muscular arteries are going to lead into arterioles. Arterioles are very thin-walled. They range quite a bit in diameter, but they're only going to have a few layers of muscle cells. The tunica media is going to be much thinner. Remember that the left ventricle is very powerful. It pushes blood out to the systemic loop of the body. The aorta receives a lot of blood under very high pressure. Because of this, it has a lot of elastic fibers. The tunic media, consisting of smooth muscle, also contains these elastic lamellae, which are just sheets of elastic fibers. Elastic arteries are also referred to as conducting arteries. They conduct the blood away from the heart. The blood that leaves the heart and goes into the aortic arch is under a lot of pressure, and it expands that conducting artery. Remember, we have lots of stretchy material within the walls of the aorta. The aorta will expand slightly, and then that stored elastic energy is going to cause it to recoil. And it actually gives the blood a little bit of an extra push. So the aortic arch can serve as a pressure reservoir. Muscular arteries don't expand the same way that conducting arteries do. They don't give the blood that extra push. Instead, they distribute the blood throughout the tissues. Here we can see the tunica media, the muscle layer of a muscular artery. These muscle cells can contract, and that can change the size of the lumen to some extent. Notice within the tunica media, we don't have all the elastic fibers that we saw within the aorta. The blood vessel on the right in this diagram is an arterial. In fact, you can even see some red blood cells in the middle of the lumen. Notice that we have several layers of muscle cells, but not nearly as much as we would see in full-blown arteries. Now let's take a look at capillaries. And there are a few different types of capillaries depending on what part of the body we're talking about. Remember that red blood cells don't leave the blood. They're traveling through tubes all the time. They don't leave those tubes. White blood cells, on the other hand, are a different story. Here we're seeing a continuous capillary. We'd find this in the nervous system, the lungs, skin, and muscle, so throughout much of the body. We have endothelial cells, which are squamous, and we have just a single layer of these cells. Surrounding that, we have a basement membrane. So these are very, very small vessels. In some cases, red blood cells have to squeeze through one at a time. These are microscopic structures. Next, we have fenestrated capillaries. Fenestra means window in Latin and also in French, actually. A fenestration is a hole or pore. We find this type of capillary in areas where there needs to be rapid exchange of materials, including, in some cases, some fairly large molecules. So we see them in the kidneys, because at the kidneys, we need to get rid of waste products that may be fairly large in size. We see them in association with the small intestine. The small intestine is absorbing food molecules, and they can come into the bloodstream more quickly with these fenestrations. Choroid plexuses are sites where cerebral spinal fluid is created in the brain and spinal cord. This fluid is going to contain some large proteins. So again, these pores are quite useful. And some glands. So glands that produce hormones, for instance. Hormones are fairly large molecules. It would take some time to move them into the cells of the endothelium and then move them out the other end. So instead, they pass through these holes. Lastly, we have sinusoid capillaries. These capillaries are full of big gaping holes. The holes are big enough to allow red blood cells to move in or out of the vessel. And that's why these holes exist. So we find these capillaries in the liver. Used up red blood cells will pass through these holes into the liver and they'll be gobbled up and broken down by macrophages. We find them in the bone marrow and also in the spleen, which are sites of red blood cell production. So the red blood cells have to get into these vessels by passing through these big holes. Remember, red blood cells can't wiggle between the cells, unlike white blood cells. White blood cells, if they get to a site where they're needed, they can leave any of the capillaries by opening up gaps between the cells of the endothelium. But red blood cells cannot do that. Here we have a typical capillary bed stretching between an arterial and a venule. Note that the arterial is very thin-walled, and in fact there's only a single layer of smooth muscle, and it doesn't even cover the whole vessel. There's even less smooth muscle on the venule. But there are enough smooth muscle cells to constrict those vessels and limit blood flow. Between the two vessels, we have one slightly larger vessel that's known as a thoroughfare. And the thoroughfare is the fastest route of blood flow between these two vessels. Blood can be shunted off to the smaller capillaries to feed oxygen and nutrients to all the cells and pick up waste and carbon dioxide. But if the blood is needed elsewhere, little sphincters, little rings of smooth muscle, involuntary muscle, can contract and limit blood flow to that thoroughfare. Here we can see this in action. So those sphincters have contracted and they've limited the amount of blood that can flow through most of the capillary. Most of the blood is now taking this thoroughfare channel, the quickest route between input and output. This is happening all the time. So capillary beds within your body will open for a brief period of time and then shut down and then open again, and they'll do this as needed. In fact, if you could somehow open all of these sphincters, you wouldn't have enough blood to fill all of those vessels, and your blood pressure would drop, and the ability to move the blood, of course, would be greatly diminished. This is what happens during physiological shock. If you go into shock because you're in a car accident or you witness something traumatic, your body goes into fight or flight mode, and one of the things it does is it tries to redirect blood towards your muscles because your muscles might need that. But in shock, it opens up too many of these sphincters and your blood pressure drops because you simply don't have enough blood to maintain pressure within all of those tubes. This is also an important feature of temperature control. So let's say you step out on a very cold day. You might get rosy cheeks because blood flow is being increased to the skin to prevent the skin from freezing. But if you stay out too long, the opposite will occur and your body will redirect blood away from the skin in order to not lower your core temperature. Once that begins to happen, then you are at danger of getting frostbite, of actually freezing and damaging the skin. Now that we've looked at arteries and arterioles, let's have a look at veins. Again, the key thing that differentiates the two is that arteries need to deal with high pressure blood and veins do not. Veins and arteries are similar in structure in that they both have those three tunics, but the walls of veins are much thinner. We have a much thinner layer of muscle, and we don't have those thick elastic layers that we saw in arteries. Again, we also have valves. These pictures show actual large veins that have been sectioned. So we've got a transverse section on the top, and we're looking down into the vein, and we're seeing two cusps of a valve. In the lower picture, we're looking at a longitudinal section, and we can see those cusps of the valve that would catch fluid. So in that diagram down the bottom, the blood would be going in this direction. Any blood that flowed backwards would get caught in these little cups and cause those cups to fill and snap together. Let's take a look at the diagram on the left. Now imagine we have blood between these two valves and we compress the vein. We squish it. Maybe it's being squished because muscles nearby are contracting. What's going to happen is the blood is going to move or attempt to move in both directions. However, the blood that goes down is going to pool in those cups and it's not going to allow the blood to move any further. The blood that's moving up, on the other hand, can move between the valves and push the valves apart and flow onwards. Venous blood, the blood that's in veins and venules, moves slowly. It's not under pressure. And for that reason, at any one point in time, about 2/3 of your blood is being stored in veins and venules. It takes quite a long time for the blood to travel through those veins and venules, much longer than it takes to travel out to the capillaries through arteries and arterioles. Because the blood entering a capillary bed from an arteriole is under pressure, fluid is going to be forced out of the capillary. 15% of the fluid that's forced out into the interstitial fluid, into the spaces between cells, is not recollected by the venules on the other side of the capillary bed. That fluid becomes lymph. Lymph is slowly collected by a system called the lymphatic system, which is going to be our next topic. That fluid is eventually dumped back into the venous blood flow. But before that happens, that fluid is passed along these lymph vessels and through lymph nodes. Lymph nodes are almost like little filtering structures. They're kind of maze-like inside, and it takes a while for the fluid to move through them. As it moves through, any debris or foreign cells, or maybe even cancer cells, will be gobbled up by macrophages. As blood moves away from the heart, it's going to pass into narrower and narrower blood vessels. As it goes from arteries to arterioles and then onto capillaries, the surface area on the interior of those vessels increases. The surface area to volume ratio is going to increase because, of course, the volume of the blood is not changing. It's just being forced through smaller and smaller passages. On the top graph, you're seeing this increase in area as we move towards the capillaries, and then a drop in the surface area as we move away and blood is collected and moved back to the heart. On the bottom graph, you're seeing that the velocity is going to drop dramatically as blood is forced into these smaller vessels. Because there's more surface area, there's more resistance, that's going to decrease the velocity down to almost nothing by the time we get to capillaries. The pressure of the blood is also going to drop. Now on the other side of the capillary bed, blood is going to be recollected into venules, passed into veins, and brought back to the heart, and you can see that the velocity of the blood increases as it gets closer to the heart because the heart is generating negative pressure. It's generating a pull or suction. But the rate at which blood flows through veins and venules never reaches the same velocity that we see in arteries and arterioles. Remember that a systole is a contraction. When we measure the systolic pressure of the blood, what we're measuring is the amount of pressure that was generated by the contraction of the ventricles. And typically we're only concerned with the left ventricle. Let's say that you measure your blood pressure in your arm. The vessels in your arm are, of course, part of the systemic loop. They have nothing to do with the pulmonary loop. So we're measuring the compression that was produced by the left ventricle that forced blood out into the arteries and arterioles of the systemic loop. As blood gets to the arterioles, it starts to be physically impeded. Before our next beat, there's still going to be a little bit of blood that hasn't made its way through the arterioles, and this results in kind of a base pressure. That would be the diastolic pressure. The diastolic pressure is the blood pressure that we can measure between contractions of the ventricles. Let's take a look at those graphs again. The top graph shows the relationship between surface area and blood vessel size. So surface area increases as we go away from the heart into the capillary beds, and it's going to decrease as we leave the capillary beds and go back to the heart. Velocity is shown in the second graph, and it's going to drop as we go towards those capillary beds, again because of resistance. The bottom graph shows changes in pressure. Note that there's a general trend here. The pressure is going to decrease as we get into arterioles and into capillaries. Again, this is because those small vessels offer a lot of resistance. By the time the blood has passed through the capillary beds, it's lost almost all of its pressure. The pressure within the venules and the veins is negative pressure generated by the atria, drawing in blood to the heart. Now notice on top of that trend within the arteries, we have this wave-like function. We have peaks that are systolic, and then we have these low points that are diastolic. The systolic pressure at the top of each of these waves is caused by the contraction of the ventricles. The diastolic pressure, which is the low point of these waves, occurs in between contractions of the ventricles. Now, if we were to measure blood pressure, there's a few ways we could do that. There's a few different units we could use as well, but traditionally we use millimeters of mercury. Hg is the atomic symbol for mercury. Think to the old school thermometers, the ones that have a glass vial that contains a silver liquid. That silver liquid is mercury. Mercury will respond to changes in temperature because that changes the pressure within that vial, and the mercury will raise or lower. You can use mercury to measure changes in atmospheric pressure in a barometer, for instance. So we use millimeters of mercury as our unit here. For a normal, healthy person at rest, their systolic pressure is 120 millimeters of mercury and their diastolic pressure is 80 millimeters of mercury. We're going to spend a bit of time looking at blood pressure and what determines blood pressure. Blood pressure is an important indicator of the overall health of your cardiovascular system. If your blood pressure is really high, that's likely caused by increased resistance within the system. That can be very problematic. The smaller blood vessels, the arterioles for instance, might actually rupture due to that increased pressure, and that can damage tissues. If you have very low blood pressure, that can be a problem as well. Even if it's slightly low, you may be more prone to fainting and passing out. So with low blood pressure, tissues may not receive sufficient oxygen or nutrients because they're not receiving sufficient blood flow. Okay, so what determines blood pressure? Well, let's start at the beginning. Blood pressure is generated by the heart. Cardiac output refers to the number of mils that are pushed out of your left ventricle every minute. Now notice we're going to focus on the left ventricle, and the reason for that is that if we're going to measure blood pressure, we're typically not doing that through the pulmonary loop. We're measuring it somewhere along the systemic loop, like somewhere on your arm, for instance. Keep in mind, though, that the right ventricle is going to push just as much blood. It doesn't push it very far, though. Remember, the pulmonary loop is quite short because the lungs are right next door to the heart. Okay, so the cardiac output, again, this refers to the volume of blood that's pushed out of the left ventricle every minute. There's two parts to this. We have the stroke volume. That's the actual volume that's going to be pushed with each contraction of the left ventricle. And then we have the heart rate, which is the number of times that the left ventricle is going to contract every minute. To determine the stroke volume, what we do is we take the end systolic volume and subtract that from the end diastolic volume. The end diastolic volume is the maximum amount of blood that we find within the left ventricle. So this is when the ventricle is completely filled. This is after the left atrium has completely contracted and pushed its contents into the left ventricle. The end systolic volume is what's left in the left ventricle after the left ventricle has contracted. So not every single drop of blood is going to be squeezed out into the aorta. There's going to be a bit of blood left behind. So for an example here, for a healthy, hypothetical 70 kilogram person, we have a stroke volume of 70 mils. And we've calculated that again by taking the end systolic volume of 50 mils and subtracting that from our end diastolic volume of 120 mils. Now, if we take that 70 mils and we multiply it by the heart rate, we will get the cardiac output. So we have a heart rate of 75 beats per minute. What that means is we have 70 mils being pushed into the aorta 75 times per minute. And we get a cardiac output of 5,250 mils per minute, or 5.25 liters per minute. So again, cardiac output is a product of the heart rate and the stroke volume, and both of these can be influenced by a number of different factors. The heart rate is affected by the autonomic nervous system and by the endocrine system. Remember that the autonomic nervous system can be divided into two branches, the sympathetic system and the parasympathetic system. The parasympathetic system maintains a resting heart rate of about 60 beats per minute in most young, healthy individuals. The sympathetic system kicks in during fight or flight responses, and it increases the heart rate. Likewise, hormones like epinephrine and norepinephrine, also known as adrenaline and noradrenaline, can increase the heart rate. Fitness levels have an impact. If you're a fit individual, you tend to have a lower resting heart rate. And if you're a younger individual, you tend to have a lower resting heart rate. The stroke volume can be affected by a number of different things. First of all, the heart size. If you have a larger heart, You also tend to have larger ventricles with larger volumes. Fitness, again, is important. If you spend a lot of time exercising your heart, if you're an athletic person, you may, through your exercise, increase the volume of your ventricles. Gender can have an impact, so males tend to have slightly larger ventricles. That's not always the case, but generally people that have a larger frame and more muscle tend to have larger ventricles. Contractility refers to the force and speed at which muscle contracts. And we'll come back to that in more detail in a moment. But if you have ventricles that contract very forcefully, in all likelihood, you're not going to leave as much blood behind. And that means you'll have a higher stroke volume. Same thing with duration of contraction. If the ventricles contract a little bit longer, then you may be able to push more blood out of the ventricle. Preload refers to the amount of blood that's in the ventricle as the contraction begins. This can be related to how much the ventricle relaxes between contractions. If it relaxes more, more blood might be able to enter, and then we have more blood to push. Likewise, this can be affected by how competent the atria are when they contract. Do they completely empty? Do they push all of the blood that they could into the ventricles? Afterload refers to resistance. If there's lots of resistance, let's say in arterioles, then blood that was pushed into the aorta might not have anywhere to go. It might take a while for it to completely leave the aorta and get to the capillary beds. If there's still quite a lot of blood left in the aorta, when the ventricle contracts again, it's not going to be able to empty completely because it has to push against this blood that's already present just outside the heart. We looked at this diagram before when we looked at how muscle cells work. What you're seeing here is a graph of the amount of force or tension that can be generated by a muscle cell as a relation to its stretch. If you take a muscle cell and you stretch it greatly, you get to a point where the myosin heads on the thick filament can't bind to the actin. That's what you're seeing at the right here. Conversely, if a muscle cell is greatly contracted, the thin filaments, the actin filaments, overlap and they block binding sites between the myosin and the actin. And that's what you're seeing on the left. Optimally, we want to have a bit of stretch to the muscle. So in the middle there, at the peak of our curve, you're seeing a situation where we have the optimal binding sites available. We have lots of opportunity for myosin to bind to actin. Now, when the ventricles contract, the muscle cells shrink, of course, and that's going to occur during systole. But during diastole, what's happening is blood is rushing into the ventricle, and it's expanding the ventricle, and it's physically stretching the walls of the ventricle, and it's stretching the muscle cells and stretching the sarcomeres. Best case scenario, it will stretch them to a point where we have the condition we see at the top of our graph. So preload, that refers to the blood that's coming into the ventricle. If we have just the right volume, it will stretch the sarcomeres to this optimal condition without overstretching them. Now let's say that you have to run for a bus. Your body is going to try to increase your cardiac output. There's a few ways it can do that. One of the ways it might do that, the most obvious, is to increase your heart rate. That will increase the cardiac output because you're pumping blood more frequently. However, if your heart rate goes very high, your ventricles never have enough time to fill completely, and they also never have a chance to stretch completely. So even though you've increased the heart rate, and you have probably effectively increased the cardiac output, you've actually reduced the volume, the stroke volume. Athletes are very good at moving blood throughout their body because they have lower heart rates, even when they're doing strenuous activity. their stroke volume remains quite high. They also have ventricles that tend to have larger volumes. So all in all, they can pump more blood than most people are able to. If muscle cells have just the right amount of stretch, we can get more powerful, more rapid contractions. Likewise, if we increase the amount of calcium within a muscle cell, we can get more powerful and more rapid contractions. Remember that calcium binds to troponin, and troponin is going to move tropomyosin out of the way. Tropomyosin usually blocks the binding sites of myosin. So if we have more calcium in a cell, we get a more forceful contraction because there's more interaction between the thick and thin filaments. Norepinephrine, also known as noradrenaline, will bind to receptors on the surface of heart muscle cells. What this does is it sets off a sequence of events inside the cell that will activate calcium channels within the plasma membrane, and they will bring in more calcium into the cell. It's also going to activate calcium channels within the sarcoplasmic reticulum. There are channels that bring in calcium into the SR. They're more active, and that means the SR can store more calcium. But this whole system also affects channels that rapidly release calcium into the cytosol out of the SR. So the net result is that muscle cells that are being stimulated by norepinephrine can release more calcium and release it more quickly, so we get rapid and stronger muscle contractions. The ability of a muscle cell to contract forcefully is referred to as its contractility. So we can affect contractility by changing calcium levels, and also we can affect contractility by changing stretch, as we already talked about. The stroke volume is, of course, the volume of blood that's ejected by a ventricle out of the heart. This is affected by something called the afterload. Afterload refers to the amount of pressure that the ventricle has to create to open up a semilunar valve. So if we were talking about the right side of the heart, we'd be referring to the pulmonary valve. If we're talking about the left side of the heart, which we generally are, we're referring to the aortic valve. Now there's always going to be some blood left within those blood vessels just outside of the heart, and that blood, if there's resistance further down the line, might be under pressure, and it might be pushing back on that semilunar valve. We want the pressure that's created by the ventricle to greatly exceed the pressure on the other side of that semilunar valve so that blood moves away from the heart. In order for blood to be ejected from a ventricle, it has to be ejected with sufficient force to overcome the force of the blood pushing back against the other side of the semilunar valve. Again, this is the afterload. This is the force required to move the blood in the aorta out of the way and move it forward. This afterload, this minimum amount of force that's required, is increased by resistance throughout the system. So blood is going to meet with more and more resistance as it moves away from the heart and into smaller and smaller blood vessels. As we've talked about, surface area is responsible for this resistance. But there's other ways that this resistance can be increased. Decreasing the diameter of the lumen of a blood vessel will increase resistance. It's a lot like sticking a nozzle onto the end of a garden hose. When you put a nozzle on a garden hose, what you're doing is you're forcing that water to go through a smaller opening. That increases pressure. It also causes the water to back up a bit behind that nozzle. There's a few ways that this sort of thing could happen in a blood vessel. We could have constriction of the blood vessel caused by smooth muscle, or we could have deposits of material laid down on the inside walls of the blood vessel. That happens during arterial sclerosis. In either case, we've reduced the lumen, and that increases pressure upstream of that partial blockage. Viscosity refers to the internal resistance of a fluid to flow. It's typically caused by things that are dissolved within the fluid that are interacting with each other. If we increase blood viscosity, we also increase resistance. Finally, we have the total length of the blood vessels that are involved in the system. This is a fascinating little fact here. If you add a kilogram of fat to your body, you add an additional 650 kilometers worth of blood vessels, mostly very tiny little capillaries. So now, with that additional distance that blood has to follow, you have to make a little bit more force at the heart. Your left ventricle has to push a little bit harder to get blood that extra distance. So the three of these together are going to amount to what's known as systemic vascular resistance. Remember that blood under pressure entering into capillary beds will lose some of its fluid to the interstitial space between cells. The plasma is the fluid portion of the blood. About 15% of that leaks out into the interstitial space is not recollected immediately. It's recollected later by the lymphatic system. That fluid gives rise to the interstitial fluid. If you have inflammation of an area, what's happening is you have increased blood flow to that area. So if you cut yourself, or if you have an infection of the skin, you're going to see increased blood flow to that area that will bring additional white blood cells and platelets to help with clotting, repair, and fighting off any potential pathogens. That increased blood flow to an area of the skin, for instance, is going to bring about redness and pain. Redness because of the increased blood flow, and pain because we have pressure building up within the interstitial fluid that may trigger some pain receptors. Forcing blood through a capillary bed is a lot like forcing pressurized water through a sponge. The water that comes out the other side will have lost all of its pressure. Likewise, arterioles contain some pressure. They've lost a bit of it already through resistance, but that pressure is lost at the capillary bed, and the blood that is collected on the other side and collected into venules has almost no pressure whatsoever. As mentioned, viscosity is simply a measure of a fluid's internal resistance to flow. In this example here, you can see the blue fluid, which I believe is just water and dye, is very, very fluid. It has very low viscosity. It doesn't resist flow. Whereas the other example, which is motor oil, has high viscosity. It has high internal resistance to flow. So what determines the viscosity of blood? The viscosity of the blood is determined by the concentration of materials that are dissolved or suspended within the blood. We have formed elements, things like red blood cells and platelets. They're bumping into each other, and they're bumping into the walls of the vessels that they flow through. So they're creating some resistance. We have things that are dissolved within the plasma, within the liquid portion of the blood. We have large proteins like fibrinogen. Remember, that's involved in blood clotting. We have globulins, which are antibodies. We have lipids and proteins and glucose and hormones and all sorts of other things that are found within the plasma. If you increase the concentration of this material, either by pumping more of it into the blood or by dehydrating the blood and pulling out water, you will get more resistance. One of the more obvious ways to change blood pressure is to change the volume of blood within the cardiovascular system. The volume can be changed quite readily by adding or removing water. So thinking back to that example of the garden hose, imagine that you have your garden hose with the nozzle on it and you're using it to water your garden. If you turn up the faucet and add more water, of course, you get more pressure, the water goes further. Incidentally, this diagram here is showing a pulse of systolic pressure traveling down a blood vessel. And this is what we feel as our pulse. So if you were to take your index finger and your third finger and rest them on your radial artery, like you would do if you're taking your pulse from your wrist, what you're feeling are these pulses of pressure. So how do we regulate blood volume? Well, I already told you, really. We regulate the volume of water. And this is done mainly by the kidney. So the kidney can extract excess water or not extract that excess water. We will get to the kidney eventually. A couple more topics, don't worry, we'll get there. But in the meantime, this is just a quick introduction to what's happening at the kidney. Your kidney contains millions of microscopic structures known as nephrons. Blood comes into each nephron and goes into this kind of ball of capillaries known as a glomerulus, and it comes in under a lot of pressure. The fluid is going to seep out and it's going to be collected by this capsule and directed into a series of tubes that will ultimately produce urine. There's a lot of stuff that goes on within those tubes, as we'll talk about later, but the kidney has the ability to pull out certain things that it wants to hang on to, certain salts and so on, and get rid of things. And at the same time, it can control how much water is going to be reabsorbed back into the blood. So if the kidney is very, very efficient at extracting water, that can maintain a high volume in the blood. If it's getting rid of lots of excess water, then that's going to decrease the volume of the blood. And this is one of the quickest ways to affect blood pressure. Blood pressure is also influenced by the autonomic nervous system. We have two parts to this. We have the parasympathetic division that's involved in so-called rest and digest activities. It maintains the background homeostasis of the body. If you're excited or scared or you're running for a bus, the sympathetic division kicks in and brings about so-called fight or flight responses. Your heart rate will increase, and also the contractility of heart muscle cells will increase. You end up pumping more blood per minute, and that increases your blood pressure. So keep in mind when we're talking about what a healthy blood pressure is, that 120/80 number, we're referring to someone at rest. Your blood pressure will increase if your sympathetic nervous system comes into play. Nerve cells that ultimately regulate cardiac output are found within the medulla oblongata of the brainstem. Here we have nerves that will send off signals to the SA and AV nodes. We have the vagus nerve that's going to carry parasympathetic inputs that will reduce heart rate and lower blood pressure. So the inputs from the vagus nerve maintain a resting heart rate. Then we have sympathetic inputs that travel down sympathetic cardiac nerves, and they're going to increase the heart rate and thereby increase blood pressure. It's also important to note that both of these inputs, the parasympathetic and the sympathetic, can influence contractility and the force of contraction. So these centers are not just affecting heart rate, they're also affecting stroke volume. Embedded within the walls of the aorta and the walls of the carotid artery, we have structures called baroreceptors. These are stretch receptors. When they're stretched, they fire off a signal back to the medulla oblongata. They are monitoring blood pressure, and the medulla oblongata will send out an appropriate response via parasympathetic nerves or sympathetic nerves. The medulla oblongata can also influence the adrenal gland and cause it to release epinephrine and norepinephrine into the blood. Norepinephrine and epinephrine can be sensed by receptors on the cardiac muscle cells. And as we talked about before, what these hormones do is they increase contractility of the muscle cells and thereby increase the stroke volume. Additional hormones can play a role in regulating heart rate. The thyroid, for instance, produces T3 and T4. These two hormones increase metabolic rate throughout the body, and they increase heart rate. Cations in the blood can be critical. As mentioned several times now, you need calcium in order to have muscle contractions. If we have very low levels of calcium in the blood, then we probably have very low levels of calcium in muscle cells, and that can affect the contractility of those muscle cells. Incidentally, if you have too much potassium, that can make it very difficult for heart muscle cells and nerve cells to depolarize. Now, this is a bit morbid, but potassium is sometimes used to put down animals, like cows, for instance, and it can be used to put down, well, criminals as well. In many parts of the world where they have lethal injection, the very last thing that they inject is a strong solution of potassium, and it stops the heart. Your body can regulate blood pressure rather quickly through vasoconstriction and vasodilation. Vasoconstriction is the narrowing of arterioles, and that's going to increase blood pressure by increasing resistance, and vasodilation is the relaxing of smooth muscle, which is going to increase the diameter of the lumen, and that is going to cause blood pressure to drop as resistance drops. Also, of course, we have the same volume of blood, but now it's in a larger container, and that reduces pressure also. The smooth muscle within the tunica media of an arteriole can constrict, and that will reduce the size of the lumen, or it can relax, and that will cause the blood vessel and the lumen to dilate or expand. Also, as we discussed before, there are rings of smooth muscle that form microscopic little sphincters that can contract and reduce the amount of flow or completely cut off blood flow to a capillary bed. And that's what you're seeing here. So if you're very warm, you might get kind of flush and red-faced because blood flow is being increased towards the surface. So we have more capillary beds opening up in the dermis to allow for increased blood flow to the skin and increased radiation of heat. So you're radiating out that excess heat. If you're cool, the opposite occurs and we don't have as much blood flow to the skin. Now, as mentioned before, this system can go awry sometimes. Physiological shock is where we have the opening of too many capillary beds. When that happens, blood pressure drops, and that's because we have the same volume of blood, but it's now occupying a larger space. If that system is working correctly, though, it can be used by your body to regulate blood pressure. If your blood pressure is too high, more capillary beds are opened. If your blood pressure is too low, then some of those capillary beds are closed down. The one thing we haven't talked about is how blood returns to the heart, despite there being very little pressure in veins and arteries. There's no pressure pushing that blood forward. So as blood passes through a capillary bed, it loses most of its pressure, and as you can see, when it's collected into a venule at the other side, it has a pressure of only 16 millimeters of mercury. Compare that to the 80 to 120 millimeters of mercury that we see in an artery of a person that's at rest. The atria, as they fill with blood, will create suction. So they do create some negative pressure that pulls blood forward. In order for this to work, it's very important that the atrioventricular valves aren't leaking. They have to close nice and tight. We need to have that atrium empty as much as possible, and we don't want any blood flowing back into the atrium. And again, this is because there's no force behind that venous blood that's coming back to the heart. We need as much suction as possible. The blood that's returning back to the heart is helped along by contractions of skeletal muscle, which we'll talk about now. Some of the major blood vessels of the body, especially in the legs, are found between powerful skeletal muscles. When you move around and flex those muscles, they squeeze on these veins and they push blood back towards the heart. Now remember that veins have valves in them. And the reason they have valves is because, again, there's almost no pressure in these vessels. Blood is going to be squeezed. When the vessel is squeezed, blood will try to move in both directions, but it can't move down in this case because it will collect in the cusps and cause the valve to close. But it can move up. Even the simple act of breathing can help return venous blood back to the heart. When you breathe in, your diaphragm is going to flatten out. That places pressure on the contents of the abdominal cavity. It squeezes veins, and that forces the blood. When you breathe out, the diaphragm is relaxing, and it's increasing pressure within the thoracic cavity. That squeezes veins again, and again is going to move the blood. Once again, the valves are arranged so that they only allow blood to flow in the correct direction. As we'll see next day, lymph, which is interstitial fluid that has seeped into the lymphatic system, is moved through the body in a very similar way. Lymphatic vessels are thin-walled and they have valves, just like we see in veins. Now, this is a diagram here that gives you a nice overview of how blood pressure can be increased, and we've talked about almost all of these already. But just looking at a few that pertain specifically to veins. If you increase the volume of blood within a vein, that will increase the pressure. If you constrict veins, that will increase the pressure. And again, that's because we have the same volume of blood, but it's in a smaller container now. And also, if you increase the activity of muscles that constrict veins, that will help bring that fluid back to the heart. So in a number of older people, for instance, if they sit for a long time, they get swelling of the ankles. And what's happening is the venous blood is not returning all that effectively. It's pooling in the extremities. And that causes excess fluid from the plasma to seep out into the interstitial space, and that causes swelling. If they get up and walk around, then we're going to be using muscles to squeeze the veins and also squeeze the lymphatic vessels and move that fluid up back towards the heart. And here's our terminology.